Recording this.
All right.
I guess recording then. All right. Thank you very much for that kind introduction, Jen Hong, and for inviting me to speak at this series, Metabolomics, a Potent Exploratory Tool in Biomedicine. As Jen Hong said, he and I are friends going way back. Jean-Paul, J.P. Kovalik, trained extensively here at Duke and actually completed 2 fellowships, one in endocrinology and one in nephrology, and I had the pleasure of being in the lab next door to his at that time. So JP and I have a lot of good tall tales from the trails about our work together in the lab. I want to talk to you today about a fairly simple and inexpensive and older technology, gas chromatography mass spec, and I'm of the, I think, minority view that it's an old technology, but it's still a good one. I'm just going to describe the technology, keep this very simple, and maybe do one example from our recent work in gestational hyperglycemia. There are some of my disclosures. These slides are in the hands of Jen Hong and JP and Chris, and there's no unpublished data in here, so please feel free to share the slides. I was asked by Duke NUS to put in this statement that this is being recorded and that they reserve the rights to use the materials for scientific or commercial purposes. I like to start off with acknowledgments so that they don't get shortchanged at the end. This is our metabolomics group. For leadership, the director of our institute, Chris Newgard, I think is pretty well known in Singapore. I had previously worked with him in industry on an artificial pancreas when he was faculty at University of Texas Southwestern Medical Center in Dallas, and I was off in biotech. Deb Muyo is a senior faculty member here who also has helped to build up the lab. We're going to talk about targeted metabolomics and untargeted or exploratory metabolomics. More about that in a little while. Olga leads the charge on targeted metabolomics. She took over leadership of the lab from me about 10 years ago. She and Bob Stevens and I have been here since the beginning. On the exploratory metabolomics side, Mike and I and Adam and Demetrius are carrying that torch presently. Much more recently, Deb Muyo has built a very strong metabolic flux program here with Scott and Guofang. And we also have a good proteomics program that's rapidly growing, but they've been heavily subscribed since they opened the door, so I'm almost afraid to admit that they exist because they've already got more work than they can do. Wonderfully, 7 of us on the staff, of the staff scientists and faculty, are on Dave D'Alessio's adult endocrine faculty at Duke. I don't know about you all, but my life always goes a lot better when I have a wonderful boss like Dave. We're at an institute here in downtown Durham, away from the main Duke Med School campus. It's built in an old tobacco warehouse from the early 20th century. And Dave has a series of labs here in our building as well, which really helps us to collaborate. This Duke Molecular Physiology Institute I mentioned, our mission statement is to use integrated, that's a key word here, multi-omics and physiologic profiles of the big chronic human diseases to develop new disease detection strategies, novel therapies, and most of all, insights into disease mechanisms. Speaking from the metabolomics lab, I think a lot of our best work has been done when we really get down and get mechanistic. So up and down the halls of this big red brick tobacco warehouse that's been refitted into a wonderful biotech facility, we have people doing traditional metabolic research in cell culture and animal models, mice, rats, zebrafish. We'll be talking in our time together today about metabolomics. We have plenty of folks on the computational biology and statistics side, very strong program in genomics and a growing effort in epigenetics. And we have plenty of clinicians, many of whom are also, or all of whom are practicing physician scientists with very heavy strengths in endocrinology, as I mentioned, cardiology, rheumatology, and some other areas of internal medicine. So what the heck is metabolomics anyway? Well, it's a little glib, but in the Encyclopedia Britannica, Chris and I define it as the study of metabolites. There are 3 major hardware technologies that are generally used today. We chose not to use NMR very early on in our history, so we're reliant on the other two. Either separate the molecules in the liquid phase by liquid chromatography mass spec, LCMS, or what I'm going to be speaking about mostly today, somehow make them volatile and separate them in the gas phase before you put them into the mass spec. I know that we've got some serious mass spectrometrists in the audience, and I want to point out that I'm restricting myself today to electron ionization with just single quadrupole mass filters, very inexpensive, simple, robust instrumentation. So there are two flavors of metabolomics. Targeted metabolomics is very quantitative, it's very precise, it's very directed. We use very extensive and expensive use of calibrants and heavy isotope labeled internal standards and longitudinal quality control measures. When Olga Okayeva gives you 15 amino acids, or Jianhong Qing, there at Duke NUS, every single well on that plate has 15 stable isotope internal standards mixed with your samples of serum or what have you. There are calibrants run at the beginning and end of each plate, and the end effect of going to all that trouble is that we can thaw the same serum, plasma, whatever, a few years later and get very much the same results. So from the beginning, we've been a little bit unusual that we've been strong in quantitative mass spec, and from their beginning, the same has been true there at Duke NUS. In the present series of talks, which I understand you can get available on YouTube, Jianhong spoke about this targeted and untargeted metabolomics last week. And 1 week from tonight, which will be midnight my time, but I want to stay up and hear JP talk. He'll also be talking, I think, focusing on targeted metabolomics. And then you guys have my slides that I'm using right now. I know sometimes I speak a little quickly. I've got an American vernacular accent, so feel free to get my slides from today. I'm not going to go through all of our targeted modules that Olga and her team, I've worked in that arena a little bit, but not very much in the past decade. We're probably best known for the combined and affordable panel, which you also have available in Singapore with Jianhong's group, of these 15 amino acids in a group of acyl carnitines. Acyl carnitines, many of you know, carnitine is a non-protein amino acid to which fatty acid tails and other things that are being digested get adducted to as they move in and out of the mitochondrial membrane. They find their way out into circulation, so you can actually find them in circulation and get a lot of insight into a person's state of fatty acid metabolism, ketogenesis, breakdown of some essential amino acids and things. Often these two, coupled with just standard clinical chemistries on a Beckman clinical analyzer, things that you clinicians order every day in a metabolic panel. When we have a big complex data set, often we'll look at these first. Oh, is it a starvation phenotype? Is it ketogenesis, some metabolic crisis? Then we'll look at those to look at central fuel metabolism in fats and amino acids, and only then look at the rest of the data and any anthropometric measures or traditional physiologic measures we have on those animal subjects or humans or what have you. So untargeted metabolomics is a little bit more of the Wild West. I'd say most labs in the world start off doing this, where we initially had our greater strengths, and to this day have our greater strengths in the quantitative targeted work. It can be a great discovery tool. The most fun I have on the job often is when we find things we weren't looking for that catch our interest. But we also drill a lot of dry wells as well, to use an analogy from petroleum exploration. And large studies, we're wrestling with several of them across the hall tonight, can be confounded by drift in the instruments. NMR can be very stable week to week, not so with mass specs. With mass specs, the critical parts of the detection in the instrument are every day getting dirtied by all the slimy biologic samples that you're sending their way. So we have a lot of quality measures on that. You can invite me back another time to give a whole talk on that. In Americans, children, when you're outdoors camping with the children, we have a children's song that says, "The bear went over the mountain to see what he could see." That's untargeted metabolomics. Unfortunately, in the song, the only thing the bear could see was the other side of the mountain. So we, especially in little pilot studies, we have a lot of these that are unproductive, as I mentioned. So this has become something of an obsession of mine for about the last 13 years. Mike and I have been working to develop GCMS as an untargeted methodology. We started off with protocols in a library, a retention time reference library, more on that in a little while, that was developed by senior leadership at Agilent Technologies, funding work at Dr. Oliver Fien's lab at the University of California, Davis. Oliver had trained originally as a botanist at Gulm in suburban Germany at the Max Planck Institute for Plant Physiology. He's the one who coined the word metabolomics around the year 2000. And UC Davis to this day is one of America's great centers of metabolomics learning. And they're also good food chemists and good wine chemists, about the best we've got. So we started off with the version of the Agilent Davis library that had 700 analytes, and we've been busy adding to that ever since. I was actually the technician on nearly all of these. It's up to about 1,300 metabolites now, and I have hundreds more where we've run the standards, but I haven't just sat down and put them in the library yet. So for a retention time locked library for electron, ionization mass spec, it's one of the world's largest such libraries. And our team working on some gestational hyperglycemia projects we'll talk about later with Dr. Denise Schultens, who's a statistician at Northwestern. We've developed a mixture model in the R language environment. We've made these programs publicly available on the web. You'll see them in our publications that are able to deal with targeted metabolomics, untargeted metabolomics, metadata of many kinds, and also be able to deal with problems inherent in untargeted analyses, such as the null cells. What if you got a non-detect in one of your samples? So Denise has been the wind under her wings on that, working out of Chicago. I would say that we've made several attempts that have stalled on untargeted LC-MS. We've got some new instrumentation on that now. I'm committed to helping us get an LC-MS untargeted program off the ground, but we don't at present have a general purpose untargeted LC-MS program. So that's maybe our one big blind spot in the traditional areas of metabolomics. I'll go back and say, fortunately, my son taught himself mass spec when he was in graduate school, got a job at Agilent, and now, just randomly by chance in recent years, he's working on the same instrument we've got. So he's 33 years old now. I've been doing stuff for him for 33 years. So now I get to call him up when I'm a confused old man, and he helps me out on that quadrupole time of flight Agilent LC-MS instrument. Dad, you're looking at the wrong part of the screen. Concentrate on the upper left. Anyway, so it turns out that today's the 17th anniversary of our lab on your side of the international date line, so wish us happy birthday. We've collaborated very broadly in biomedicine, but we've always maintained a strong focus, at least I have, and a lot of the people I work most closely with, on diabetes, obesity, cardiovascular disease. Thankfully, I'm getting pulled more into kidney disease these days because that untargeted GCMS is really good at the urines and the uremic retention solutes. So yeah, wish us happy birthday. I stopped counting about a decade ago when our number of collaborators around the world exceeded 300. Obviously, many of those are small pilots that don't go anywhere. People test out our phenotyping capabilities and don't find them to be of use to them, but we've developed some very strong relationships, and some of the most fun ones are when we start helping people when they're fellows or graduate students, and these develop over the years to where we get the pleasure of being involved in their career development to the higher ranks in academia. A lot of labs around the world are afraid to do big studies. I'm afraid to do them too when it comes to non-targeted, but we have ways of dealing with that, and our largest clinical studies approaching 10,000 specimens. Now, I know for some of you genomics people that might not be particularly impressive, but in the metabolomics world, that's a lot of samples. Number of publications is probably over 200, certainly over 300 by now. We haven't done a count lately. Our model is very collaborative. We're very different than some other metabolomics labs that we respect and that we work with here at Duke. Our model is we don't like to get involved with the study unless our level of contribution intellectually can rise to that of co-authors. So that $250, $300 count just includes the ones where scientists working at the bench in the lab are co-authors. It's very egalitarian. Every one of us works at a bench with a pipette and a wrench. Some people in the lab object to working on monkey tissue because they can't always guarantee they won't have those herpes viruses. So when you see Chris Newgard, me, and Bob Stevens, all of whom are over the age of 60, gowned up in biohazard gear grinding up monkey tissue, you'll know what that is. I've got samples running across the hall tonight. It's after 8 p.m. tonight, and I'll be checking on those urines and sera before I go home tonight. We're up to the lucky number of 13 mass specs now. We really enjoyed our early work over a decade ago with NUS, Duke NUS, and other institutions in Singapore. This was a favorite early paper of ours in Diabetologia. I'm sure you recognize some of the co-authors. What really struck us as being unique about Singapore, I can't think of another industrialized society like this, is that you have a number of very distinct populations of different ancestries, notably Chinese, people of South Asian descent, also Malays, if I can call myself a Caucasian, white people, whatever, living alongside each other, eating similar diets, having similar levels of activity, but having very different burdens of the great metabolic diseases. So we were convinced very early on, you didn't have to tell us twice, that Singapore was and is a fertile ground for discovery in the great metabolic diseases of humankind. So the initial tech transfer process that Jen Hong was active in and was describing a little while ago lasted for 2 years, starting in January of 2010. They started off as a partial phenocopy, if I can say that, of our lab, but the two labs have since diverged and developed unique and complementary skills of their own. We maintain a very strong and sharing collaboration. I enjoy nothing better in my job than hosting their lab visits. It's not unusual at all for people to go to either site and stay for weeks at a time working at the bench, transferring technology. Recently, we had the pleasure for the entire month of September 2018 and a little bit before and a little after, we had their entire scientific staff and faculty here at one time or another, and got to have a reception with lots of Singaporean foods, including the famous durian. And we had their help of the Singaporean experts helping out myself and people from our lab, picking out a particularly fragrant durian for us to have at that reception we had with them. People brought their children, people brought their parents. It was a real, September 2018 was a new high point in our relationship, I'd say. So we have 13 instruments, as I mentioned. There's the time of flight QTOF that I want to develop with the team. Non-targeted LCMS. It won't be our first attempt at that. And again, my son's helping me out as an unwilling and unpaid technician on that when his dad gets stuck. But I mostly want to talk about GCMS today. So how the heck does GCMS of metabolites work? It's a very simple and stable technology. It's been practical since the early 1970s, meaning it had pretty much arrived in its modern form, lacking a lot of automation and standardization. And people in hospitals, medical centers began to be able to purchase systems that you could use off the shelf instead of having to make all your own stuff and coat the linings of your own columns and everything. And my goal today is very simple. I want to keep this short for a broad audience, talk about the technology a little bit, and talk about one of our studies that's ongoing in gestational hyperglycemia to show you one of the things we do with it. Unfortunately, you have to extract the metabolites. Nearly everything that you and I study in intermediary metabolism is too polar to stick in a gas chromatograph column and heat up in an oven and have it vaporize and fly. If it has oxygen, if it has sulfur, if it has nitrogen, it's generally going to be pretty polar and going to be difficult to do good, consistent GC on it. So we have to extract them and dry them down and do a couple of derivatization steps, which complicates and, you know, and lengthens the trouble we have to go through on that. But they're reproducible processes. The GC column, and I've got one right here in my hand. It's just a hollow fiber of silica. It's got like that orange polymer polyimide you use in bulletproof vests wrapped around the outside to make it so we can handle it and run it up to high temperatures without having it fall apart, but it's just a hollow fiber. So you're flowing helium through there, you inject the sample into it in an oven. The sample condenses in the first few centimeters in the walls of that hollow tube. You flow dry helium or hydrogen, if that's what you prefer, through there and just start heating that oven up. And the light stuff comes off early and the heavy stuff comes off late. It's not strictly distillation, but it's something a lot like a distillation, the way we're using it here. And in the protocol that we're doing tonight here in the lab, derived from the Agilent non-targeted GCMS program, we ramp from 60 degrees Celsius to 325 degrees C, which is hot. If you have to reach in there and work on something, you can burn yourself. And then we blast them. So that's the chromatographic part where we're separating the little molecules. We hit them. Last week, Jen Hong talked about soft ionization. All of the liquid chromatographs in our lab tonight run on electrospray ionization, which is very soft. And if you want it to, with good luck and the right molecules, you can get the intact molecules out. Not so with the way we're using GC in this. It's a very hard ionization technique. So the bad news is you have to really blow them apart. This is simply a little coiled filament. We look at these, we get them dirty, they warp, they whisker, they get destroyed. It's just a little coiled filament of the two highest known melting metals in the universe, tungsten and rhenium. I've got an old worn out one here. I'll turn it toward the camera. You probably can't see that, but there's a little filament on there that Thomas Edison would recognize. And when we put electricity through there, it glows like a little light bulb. That's when you know that it's on. So that blows apart the molecules. We have to put a charge on them to be able to sort them out in the mass spec filters. We're actually making radical cations here. In the protocol I'm describing, we scan from 600 down to 50 and go back and scan from 600 down to 50, roughly from about the weight of derivatized cholesterol down to that of lactate. So I said I spent a lot of time working on libraries and obsessing about them. I had a lot of help from Agilent learning how to do this. So how it works, it's an orthogonal peak annotation. Fortunately, compared to liquid chromatography or capillary electrophoresis or running polyacrylamide gels, things most of you have done, we can get them to come off at exactly the same time day after day if we're paying attention and modifying certain pressures day to day to day. We've gotten a lot of practice at it. We can get these things within a few seconds. We can make lactate behave and come out right where we want it. So we know the time it comes off the column. I said the bad news is we're blowing these molecules apart to ionize them, which is pretty crude. But the good news is those fragmentation patterns are very reproducible. You can go back to ones in some of the big libraries we buy from the government and Wiley publishers, and it's remarkable how well our mass spectra match up to those of people that were working back in the 1970s. So as an example, here's capsaicin. I know Singaporean food is not typically that spicy, but that's the main hot element in hot peppers. And you see, if I run the pure standard of capsaicin, it comes out right here at 23.93 minutes with the characteristic heavy ion. Here's the chromatogram showing all the ions together. Here's the extracted mass spec right there at the peak at 23.93. And we see we've got a heavy fragment at 209.1 mass to charge units. We love heavy fragments. They're often very characteristic. We don't like light fragments much at all. So, okay, so now let's put some hot sauce in the instrument. Here at the right, we've taken 100 microliters standard aliquots of just hot sauce off the shelf. In America, we have an Americanized Mexican fast food chain called Taco Bell. Their mild sauce is this black peak down here, and you see it barely comes off the baseline at all. Their so-called fire sauce is the red one there. I'm an Arizonan. Don't try to tell me that's hot. Louisiana Tabasco sauce, which you might know. The same one, obviously, the capsaicin content's very high. And I only regret that I didn't have any Singaporean chili crab. I haven't seen it for sale here yet in Durham, North Carolina, that I could have run alongside that. So you get the idea. You know, we think we know how we're annotating the peaks correctly because we've got a retention time that's reproducible and a mass spec fragmentation pattern that's reproducible. So what's it good for in general? More than half of the GCMS instruments that are sold worldwide are used in the petrochemical industry. You can dilute them and shoot them. Don't have to go through all those horrible derivatization reactions I described for, you know, polar metabolites. Flavors and fragrances are very important. Burt's Bees, which is a famous American beeswax cosmetic company. I think the chapstick in my pocket is from Burt's Bees of Durham, North Carolina. They've got GCMS instruments the same as ours in the lab. The fragrance industry, perfumes, for instance, you just dilute them and shoot them. We often do our teaching with perfumes. When the Juvenile Diabetes Research Foundation meets here in this building, and later in the meeting when the bankers and the serious principals and, you know, the suits want to get together, if they have their children with their insulin pumps, typically to entertain the children during the heavy business meeting, we take them in the lab, we get their mom's perfume, we dilute it and shoot it, and we figure out what their mom's perfume is made out of. And they know exactly what we're doing. Their kids are sharp, and GCMS is pretty simple. It's still very heavily used in monitoring environmental pollution, pesticides and herbicides, and lots of other food adulteration scandals often are sussed out by people working with GCMS. Crime labs, all the crime TV shows will have often thermo or adjuvant GCMS instruments right there in the on the television set of the crime lab show. For example, figuring out what types of fire accelerants might have been used in an arson crime. GCMS is still used for probably most of the drugs of abuse work. Some of that's going over to LCMS use, but of course, we're interested in using them for human metabolites in our lab. You can go as light as things with breath condensates and things as heavy as, say, the lighter cholesterol esters. Let's talk about how this found its way into metabolic profiling. Who were the early adopters of GCMS in human medicine? Now, physicians have been long interested in urine. I mean, the element phosphorus actually came from, because urine looks gold to some people, alchemists trying to derive gold from human urine came up with the first preps of phosphorus that was discovered. But the physicians have been smelling and looking at the colors and tastes of urines way back since well before 1500, when this one was 1506, when this one was published. You know, they saw a condition where people made lots of urine passing through big volumes of it in polyuria. It had a honey sweet taste, mellitus, so they named it diabetes mellitus. So perhaps not surprisingly, in the early 1970s, when the GCMS technologies began to stabilize, you can actually buy one that you can use in a clinical research laboratory. The pediatricians jumped on it immediately and made good use of it. Especially in the organic acidurias, notably the inborn errors of metabolism. GCMS fueled significant discovery of new acidurias. It's still helping to fuel progress alongside the newer LCMS technologies in that. There was a wonderful book. The British were especially good at this then and now, especially in the inborn errors. And Chalmers and Lawson's Organic Acids book still travels with me. It's still fresh reading, you know, almost 40 years later. And in this figure, they show the number of organic acidurias that were known at the time. And a lot of this work in the 70s through the 80s, that was all fueled for the most part by GC. Earlier techniques, they were using thin layer chromatography and other wet chem techs and microbial assays and stuff, just really limited what they could do. And very quickly after that, the nephrologists studying uremic toxins, I think now we would call those uremic retention solutes, jumped on very early on too. And these, both of these are of great interest to us in our lab. Our lab actually started as an outgrowth of a pediatric biochemical lab at David Millington, Bob Stevens group at Duke Children's Hospital. Duke's long been a center of that sort of biologic mass spec. I want to point out that in the early 1970s, including some work that Linus Pauling did late in his career of breath condensates by GC, they were simultaneously measuring dozens of metabolites. That's 25 to 30 years before Oliver Fien coined the word metabolomics. So a lot of people in our mass spec community here in the Durham Triangle area, sort of groan and roll their eyes when they hear that we're doing metabolomics by GCMS still. But I think it was a prophetic pioneering work in metabolism. I think it really foretold the coming of metabolomics before any other major technology had done so. And I think it's still of use. So yeah, Jen Hong wanted me to mention a few things about the gut microbiome. We have very profitable productive relationships with Jeff Gordon's group at Washington University, St. Louis. I'm not going to go into depth on that today. You can invite me back sometime, and you can look at the papers co-authored by Chris Newgard and Jeff Gordon with people from our lab on it, just to look at a couple of one particular metabolite that loans itself to GCMS. Hippuric acid is a major urinary organic acid. We'll get to hipurate in a minute. We have lots of benzoic acid flooding around our system. We put it into into canned sodas as a preservative. It's naturally free in fruits such as apples, but the great majority of the benzoate that's floating around in my blood right now no doubt came from gut microbial action on aromatically stabilized compounds. We know that gut bugs can get to benzoate from purines, big time from phenylalanine and tyrosine, flavonols, lots of those plant phenolic compounds. And the little microbes are very lazy. We've come to appreciate that in working on these compounds over the years and what they become when they get into the human body. The microbes will take the aliphatic stuff that's not resonance stabilized, that is, it's not the aromatic rings, and they'll chew it down. And when they get to one carbon in this case, or 2 or 3 carbons away from the aromatic ring, they just let go of it because they're too lazy, or it's not energetically favorable for them to put the energy in there to break apart an aromatically stabilized ring. I used to teach organic chemistry to pre-medical students. You can avoid the holiday rush and start hating me now. Anyway, so we have lots of these little aromatic compounds with 1, 2, or 3 aliphatic compounds sticking off of them in the gut microbiome story. And what the human does with benzoate is glycine and acyltransferases and standard phase 2 detox in liver, kidney, and elsewhere. Those of you that have studied pharmacology, cancer biology, done detox reactions, this glycine conjugation is very familiar to you. We have lots of glycine conjugates in urines and human, even more in rodents. I'm still busy adding these to the library. Our library, I've had to synthesize a few of them ourselves here in the lab. But what the main fate of benzoate in us is to make hippuric, hippo, horse, uric, urine acid, which is nothing more than N-benzoyl glycine. We can secrete hundreds of milligrams of hippurate in our urine a day. In the urines that I'm running across the hall tonight in a study of exenatide biology, often the hippurates and the derivatized ones with other things decorated around this ring are some of the biggest peaks in the field after urea. So benzoate has actually been known for a long time. Nostradamus, who's very much an alchemist, distilled it from a European plant named it benzoin. We would recognize his prep today as pretty pure benzoate. But at the very beginnings of organic chemistry and physiology out of alchemy, Justus von Liebig, who's an academic ancestor of mine and probably many of the people who were in on this call, there weren't that many organic chemists back then, he identified hippuric acid and characterized it from horse urine. And then they fed the horses benzoate and saw that they had a big increase in hippuric acid. And then they ate benzoic acid themselves without approval from an institutional review board and saw that their own urine was very much enriched in hippuric acid. So that's 1842, my friends. So some of the beginnings of human physiology research. So it's a major human organic acid. And I keep seeing all these good studies that it's generally lower in urine of obese and type 2 diabetic humans and rodents. but nobody yet has told me why. We've observed this as well. So we've got all kinds of arm-waving explanations for it. Is the diet of these two groups of people somehow different? Is it something about the activity of the gut bugs toward aromatic compounds? Is it the barrier function of the gut is somehow changed? Often in these pathologic conditions, your gut actually gets leakier. Is it something to do with the change of phase 2 conjugation in liver and kidney? I don't know, but as I mentioned before, this is one of the big ones that will often come up in studies of gut microbiome, not just plain studies of diabetes in rodents. Often seen, still not really understood. So what are some of the strengths and weaknesses of GCMS? I mentioned that the retention times are stunningly reproducible if you're paying attention. The mass spectra, it's unfortunate that they're blown apart, but the fragmentation patterns are reproducible. We can't always, sometimes we can identify an unknown within a few weeks, and some of the unknowns we've been looking at for more than a decade we've never been able to identify. It doesn't really give you an accurate mass like some other instruments, only about 0.1 atomic mass unit. It's very inexpensive. Jan Hong was telling me some of the prices there in your Southeast Asian markets. In the US, the academic price for a good developed system is about 100 to 110K, where the LC gear that we've got in the lab across the hall start in the sort of 200s and go up to $1 million. And in addition to the purchase price, you have to pay 8 to 15% after the 1-year warranty as a maintenance contract on them if you can afford it year after year. So these things add up. So we like the inexpense. It's very robust, very reliable, almost no moving parts except for the flow controllers and the little robot that moves the little sample bottles around. They put these on ships at sea. They build stripped down ones for planetary, probes to look for organic molecules on other planets. Fortunately, they don't have to have vacuum pumps because they're generally operating in a vacuum. It's really easy to learn. It's how we start teaching the new scientists. I helped teach a course on that for young physicians working in metabolic disease, especially. More on that in a minute. It's very generalizable for small polar molecules, which can be difficult. Protocol development can be very difficult in LCMS. Generally, if you bring me something new, somebody brought me some goose feces from the parking lot one day, generally within a day, we can get them extracted and get them into our protocol and get some kind of data out. I'm not saying to be optimized. Obviously, that's for just non-targeted work. Obviously, when we do targeted work with stable isotope internal standards and unlabeled calibrants, the development time can be much longer. Probably most of the money that flows through the non-targeted operation in any given year is serum and plasma. They tend to be pretty homeostatic, or you have a terrible metabolic crisis and die a horrible death. We do a lot of work on cell cultures, and especially their conditioned media are often more consistent than the cells themselves. I mentioned that we do cecal and fecal matter in microbiome studies. I'm especially proud of the work that our lab does on tissue homogenates. We've been doing that all along. Olga and her team have gotten to where you can give us a 20 milligram punch biopsy of human muscle, and they can give you consistent metabolomics on that. I like urine because it's so rich in all the small polars that GCMS is good at. I keep asking you cardiologists and endocrinologists for urine, and you keep giving me serum and plasma. Stop doing that, please. No, we really like urine. We have a lot of fun with it. Currently, and the urines that are running tonight, we'll probably report about 200 consensus metabolites. With some library building I hope to do in a month or two, we ought to be able to push that up to 250. I mentioned some of the limitations. You've got to extract and derivatize first. That's got a lot of hazards with it. Some things give you more than one peak that aren't necessarily additive. That includes aldehyde reactive sugars like glucose. That includes the amino acids. And a lot of small amines are difficult. Give us a whole bunch of peaks where we'd really rather only have one. You can't do the bigger molecules. It's really slow. Sorry, I should have said 38 minutes there on the protocol we're running tonight, so it's not high throughput. We do these studies with hundreds and often thousands of samples. We're crawling through them at 22, 30 samples a day with all the quality control measures. That's a grind, and it slows down the number of studies that I can get done or the number of new collaborations I can fire up in a year. And there are a bunch of little molecules that just won't fly in GCMS. They're thermolabile or they're very highly polar. They just sit in there and scorch. quaternary amines like choline or carnitine, guanidino compounds like arginine, Zwitter ions, sulfates like to shed their sulfates, things like ATP that have phosphoanhydride bonds. So yeah, it's got a limited range, but it's really good at those little organic acids that are so important in metabolism. So I mentioned that we treat a boot camp here, and I'm going to invite all of you to come attend if you want. Just come spend some time in Durham. It's based on GCMS. Mike and I teach it. We really go after the practicing physicians and surgeons that want academic careers, catch them earlier in their careers, medical students through junior faculty. Some PhDs also come and do this with us. They learn to tear down and clean and reassemble and passivate every single user serviceable part on our GCMS system, tune it, prep and run their own samples, analyze their own data, and write up their results. So if any of you in Singapore want to send your trainees to them, we'll put them to work. I see that my time's getting a little tight, so we do work a lot in the metabolic diseases. Pinar's been with us since she was a fellow working in pediatric endocrinology, and they've all had productive times with us. We're especially proud when we have the K Awards that were developed around metabolomics work we do. So I'm just going to give one practical example, and then I hope leave a little bit of time for discussion. This is a paper that was very unsettling to a lot of people, a lot of provocative, wasn't from our group, that came along back in 2008. The palmitoleic acid is just the cis C16 fatty acid. Humans can make fatty acid, can make palmitate from scratch, and we can make palmitate into palmitoleate with the desaturase called SCD1. So this is something we can make out of thin air, but they proposed that it's actually some kind of a hormone. They called it a lipokine. It's a simple fat that can act as a hormone with systemic effects. And in a mouse model, they observed, it's a pretty good paper, that the adipose-derived C16-1 stimulates muscle insulin action and suppresses hepatic steatosis. This paper has been much cited and debated. It's got over 1,000 citations, so just hang on to that thought. We're going to see palmitoleate again in a minute. My own work as an investigator involves doing genetics and helping get metabolomics and helping get the genetics done in the Duke part of the hyperglycemia and adverse pregnancy outcome study, which I'm going to call a HAPO. Bill Lowe's a vice dean at the Northwestern Medical School in Chicago. He's an endocrinologist who spent his career working in gestational diabetes. I mentioned Denise is a very gifted, creative statistician and data analyst. So they enrolled expectant moms worldwide, including in Singapore, that were hyperglycemia but not frankly diabetic. Initially, we did metabolomics on 1,600 newborn dyads in 4 different ancestry groups, not including the big maternity hospital in Singapore. Unfortunately, I haven't worked on those samples yet. Get a hold of me if you want to do some metabolomics together with our friends Jan Hong and JP and company. The Afro-Caribbean women from one hospital in Barbados in the Caribbean, Northern European women basically from around the world, English-speaking countries, Thai women from one maternity center in Bangkok, and self-identified Mexican-American women from Bellflower in suburban Los Angeles here in the United States. So it's 7 months of gestation. They come in fasting, 1-hour glucose tolerance test. Obviously, many other samples are grabbed, but in our lab, we've always focused on doing the fasting and the 1-hour sera on the moms, and then 2 months later, we grab the baby's umbilical cord blood. And we often run these together, 1, 2, 3, 1, 2, 3. In this initial set of studies, 4,800 sera, less than 2 dozen a day, do the math, it's a grind. We've had 11 publications so far in leading diabetes journals. I've spoken at length, I'm sure ad nauseum, about HAPO before, but there's a new paper just came out in September that I'm going to spend the rest of my time speaking about briefly. I sent the PDF of that to Jen Hong and Jean-Paul earlier tonight, and they should feel free to share it with you. An earlier finding was that a mother's BMI is positively correlated with a neonatal cord blood metabolic signature characteristic of insulin resistance and the risk of type 2 diabetes in adults. that includes both things that can cross placenta and things that can't. So it's kind of disturbing finding to us, and we've been following up on that. But in the current paper, we're looking back at that initial set of 1,600 moms, just looking at the fasting and 1 hour data from month 7 of pregnancy. GWAS in a bigger group, and the overlap was a little about 1,400. Did per metabolite analysis, fasting in 1 hour, targeted metabolomics, untargeted metabolomics, and those conventional clinical things from the Beckman, things like ketones and free fatty acids and so on, triglycerides. GWAS found 12 genetic variants in the glucokinase regulatory protein gene locus, GCKR, that were associated with maternal insulin sensitivity. And this includes a very common missense mutation. I'll just call it 326 from the last 3 numbers. This SNP is associated with maternal serum metabolites in both fasting in 1 hour with the triglycerides, and an interesting guy called 2-hydroxybutyrate, which I can give you a talk on some other time. I think this is a marker of oxidative stress, and it's coming from glutathione synthesis. It's been seen in studies, population studies of non-pregnant people, as a marker of diabetic risk or cardiovascular disease risk. And at 1 hour, oral glucose tolerance test, lactate. You can't listen to a talk about metabolomics from Duke without hearing about branched chain amino acids. The keto forms, the proximate metabolites of leucine, isoleucine, and our old friend palmitoleic acid, C16-1, came up and hit us in the face there. So overall, it looks like there's more metabolic fuels involved than just glucose. Mediation analysis suggests that the 1 hour palmitoleic contributes in part to the association of 326 with maternal insulin sensitivity, explaining about 14% of the total effect. And I'd point out to people that don't like GCMS, that GCMS was important for several of the metabolites here. We might not have as easily gotten them from a broad LCMS scan. So just kind of wrapping up here, here's the model. We're saying that C16-1, it's not at all what I expected, and we didn't want to believe it at first, has something to do with the interaction of the glucose kinase regulatory gene on mom's health and her fuel metabolism. So we wanted to bring all these, I'm speaking of the whole HAPO project, of which we're just a small part and latecomers. I was involved in a number of grants to try to bring in the moms and the kids, now that they're 10 to 15 years older, to resample them. And the bad news is we struggled. We got rejected by the National Institutes of Health here in the US and other entities I won't mention for the sake of decency. But the good news is that by the time we got it refunded, it was 10 to 15 years later. So now we've got, the data from mom as she's pregnant, the kids when they're newborn, and now we're looking at correlates of how the kids turned out. Are the kids obese at 10 to 15 years old? Is it easier on a mom 15 years later to have had a male or a female fetus and so on? So please stay tuned on that. I think GCMS is not glamorous here in my last slide, but to me it remains a useful workhorse for studying human metabolism. I think the mechanisms underlying metabolic disease are still largely a maze, and that maybe metabolomics can help us find our way. Thank you. Jan-Hong, are you there?
Yes, I'm here. Thanks, James, for the informative talk. Opening up the questions, Q&A session to the audience. Do you have any questions to ask James?
Millions. May I start?
Yes, please ask.
Yeah, this is Jens. Hello. I'm from Germany. I'm at Duke NUS, and also part-time faculty at nephrology at Duke.
Great.
Until today, I did not really know what I should do at Duke, North Carolina. I have to meet with you. That's very important. We need to meet online and as soon as possible in person. I need to renew an R01. I was thinking of relinquishing my green card, but it looks like if the last votes in Pennsylvania come in, I will not relinquish it. And then I will, and I'm serious about it. And then I indeed will will renew the R01. And I was just very surprised to meet the first metabolomic person who said, I'm interested in urine. So here's the story. I have made a balance study in 10 humans in which these two guys ate every crumb of food that was totally controlled for 205 days in a row. And they collected every drop of urine.
We do 24-hour urines. That's great. Yeah, most of what
yeah,
we do is spot. Go ahead.
So long story short, we know exactly what they ate for more than half a year every day. We know how much fat they ate, how much. Well, it's the longest balance study that has ever been done in humans. And so the first question that I have: how stable are these metabolites in the urine? The samples are, most of the samples are stored at 40 degrees Celsius, minus 40 degrees Celsius now for 9 years. There are some samples, very few. There are 2 milliliters or 5 milliliters, I think, which were stored at minus 80 now for 10 years. Nobody ever touched them. But we have something like 100 milliliters that have been stored at minus 40. What we find is that it seems to be very stable, but of course now, what is your thinking about that?
Yeah, I think that that most urinary metabolites are much more stable in frozen storage than you would think. Now, water has a measurable vapor pressure, even at liquid nitrogen temperatures, minus 199. So the ice crystals turn over, and that can bust some things up, like some sensitive, hormones and cytokines. It's been known since at least the 1950s that even in really long-term storage, asparagine will naturally shed some ammonia and become aspartate, and glutamine will shed some ammonia and become glutamate. That's very well known, and there's not really much of anything you can do about it. Because there's so much of that in, say, in human blood anyway, it's not that great of a difference. But like in the drugs of abuse monitoring by GCMS, and in 24-hour urines, like in our pediatric stuff where the kids are walking around with a jug, I've got some of these red jugs right outside, and not necessarily keeping them in the fridge. A lot of that stuff's remarkably stable. And the way we know with urines is the alpha keto acids and the beta keto acids. If we look in there, in the first sample we prep up from you, and it's got pyruvate, which is an alpha keto acid, it's got alpha ketoglutarate, which is an alpha ketoglutarate, it's got the ketoleucine, keto-isoleucine I just showed, which is an alpha, and then like acetoacetic acid, which is a beta keto. Those are all very fragile. And if somebody let the thing thaw and then refroze it and didn't tell you, those will all fall apart. So when we see those, the alpha keto and beta keto acids in urines in abundance, we go, ugh, they didn't mess it up and we didn't mess it up. So we have some things, some quality measures. There are others, but I won't drag it out from there. But yeah, we'd be happy to do a little pilot and look at that. Of course, Jianhong's got all the same capabilities there at Singapore and with Zing and all. But to be happy to write letters of support on your R01 renewal if that'd help.
Yeah, so the samples are sitting in Germany at the moment, and the question is whether we move them to Singapore, or whether we will move them to the United States. It's just, I think it's a unique balance study. This has never been done before. We are working with Manfred Rau in Erlangen, whom you perhaps know. He of course knows you.
Yeah.
Right. He of course knows you. We simply need to talk about it because I need to be a bit creative now with pursuing my scientific life in the United States.
Yeah, the fact that they're 24-hour urines instead of spot urines, which is what we mostly deal with, and we normalized the creatinine, which has got its problems, that should strengthen it. But great, look forward to meeting you.
Thank you.
Jianhong, you're muted.
Oh, sorry. Yeah, so I have 3 questions here. Let us quickly go through them because we are already past 10. So sure. First question from Zhen Xiang. What type of columns are you using and how do you prepare the samples for aqueous phase samples?
Great. So the column that we're using, and I had one in my hand a minute ago, is a standard, it's a DB5MS, ultra inert, which is a very conventional general purpose heat resistant column. Happy to send all those details on request. It's in the Agilent protocol. The way we prepare samples varies somewhat from sample to sample, even though they're all water, a lot of them are water-based. Serum, we start with a methanol crash. So we crash out the proteins with methanol, probably much as Jan Hong does for serum or plasma. Take that supernatant over and dry it down in a speed back and derivatize that. With urines, it's somewhat different. With urines, while they're still in the water phase, we do the first derivatization reaction. We extract twice into ethyl acetate and then dry down from ethyl acetate. So, and we have some other things for cell culture, but those are the two main ones. Is a direct crash for serum and plasma and an ethyl acetate extraction for urine.
All right, thank you. And the next question by Amy is, well, what she said was the Liu study was fascinating, and she's familiar with GCMS, but new to GCMS. Does the primary advantage of the technique is to detect many molecules at once to identify the correlation between individual molecules and the disease? What's the question actually? Does the can you read from your side, James? Does the
I can't. I wasn't able to pull up the chat log yet. Sorry. Let me try.
I think is the primary advantage of the technique GCMS to detect many molecules at once to identify the correlation between individual molecules and the disease.
Yeah. So we, you know, I only described the way we acquire the raw data, and I didn't go through the data reduction after that. But yes, I think that ultimately just a bunch of numbers about levels of different metabolites is not that interesting. So we've been using the free software. We work with collaborators that do data reduction in many different ways to link the results to, you know, physiologic health of an animal or person. But the free systematic software from the Canadians called MetaboAnalyst has become very user-friendly. You can teach yourself to use it in a few days. And yes, I think one of the main goals is we don't just want information, but we want knowledge about how these things interact. And then I hope in some small way, some wisdom eventually that can give you insight into moving forward. Yeah. But yeah, the first place I'd send you would be to the MetaboAnalyst freeware from the Canadians.
Right, and Chi-Min and Chun Hui Liu has almost the same question. Can you elaborate more on the metabolomics bootcamp? I think we can take this offline. Chi-Min can email to James directly to ask for the bootcamp. Sure. And Chun Hui Liu is asking whether Duke NUS has the same metabolomics bootcamp. We can come up with something. Yes, I'll be interested in that.
It's nothing fancy. There's no classroom. They just work at it. We keep an older instrument that's fine for training going, and you know, I go across the hall where everybody helps them, and they just get embedded with us for a week, 2 months. You know, however long they want to stay. But yeah, we've done it in as little as a week. Yeah, they're certainly welcome here, Jen On. But yeah.
Oh, I wish I can come over. Yeah. Hopefully. After everything settles down, after COVID is down, we'll be happy to come over here as soon as possible. Yeah, last question. So for Chun Hui Liu and Chi-Min, I will get back to you. Last question from Aaron. Do you usually use retention time blocking in your libraries, or could you use linear retention indices instead?
You could calculate your own retention indices. I'm most comfortable. I think it's stronger. Basically, I take the only standard we put in there is per deuterated myristic acid, and the trimethylsilyl ester of that we set at exactly 16.727 minutes. When I go across the hall before I go home to bed now, I'm going to go see where that, you know, that's there, and it drifts down slowly as the column ages every day. So we nudge some pressures, but yeah. You can do the conversions and, you know, run other protocols and still use those libraries by converting the retention indices. It's actually deep down in the software. It's not based on time. It's actually based on retention index. And I have equations, quadratic interpolation that I've derived that relate our retention times and indices to Agilent's, to the GOLM one in Germany, and to the one on the NIST, the giant NIST Wiley library. So I've got quadratic equations stuck up here on my wall that do just what you're requiring, that show this index versus that time. You can run ladders of fatty acid methyl esters, you can run ladders of N-alkanes, we do both. But yeah, all of those things are interconvertible if you're willing to wrestle with the resultant quadratic equations.
All right. Okay, one last follow-up question from Aaron. How many sample replicates would you recommend for performing untargeted or discovery metabolomics?
That's a good question, and there are a lot of different answers out there. At present, we're only running one injection. As it is, we're only able to get through 22 to 30 samples a day, plus all the quality measures. And if we were to do double injections, which we do sometimes, when like premature babies where the samples are very, very precious and the volume's limiting and everything, and we're the last ones in the line, we'll just re-inject those over and over till they're gone, to get the best measure. But in these giant population studies, we're just doing a single injection right now. And I'm sure we're open to criticism on that. Now, mind you, like we prep up in the big studies, I wasn't going to go into quality measures. We steal a little bit of sample from every sample in a study, make a pool, and then every day one of those pools gets prepped up and run fresh. So we inject that at the beginning, the middle, and the end of the run. So we're watching the reproducibility on those to see if we have a problem. But yeah, we're just doing single injections most of the time.
Right. Thank you, James. Will you unshare your screen? I would like to share the advertisement for next week's talk.
You want me to unshare my screen?
Yeah. Okay. Yeah. So stop share. You go
Stop
to the
share.
top. Yeah. All right. All right. So for those who are still here, we have a talk next Thursday, 11 a.m. by J.P. Kovalik. So what he'll be talking about would be how to use our use our targeted workflows in order to get the best use. So how to use them to study metabolomics in your project. Yeah, and it will be quite informative. It's a targeted talk on LCMS. Thank you very much. Thanks everyone for being here, and thanks James for the talk.
Thanks everyone for their kind comments. I've seen coming in. All right.
Thank you.
Wish us luck on the election.
Yeah. All right. Good night.
Bye.
Bye.